Good afternoon. I'm Marta Oliveira from OncoAdvanC Plus, and we are here to present a unique therapeutic option for advanced cancer. So it goes without saying that cancer is a high-impact disease. It kills per year 8.2 million people worldwide, and the number of new cases of cancer are going to increase by 70% in the next 2 decades, reaching 22 million cases per year. But what causes cancer, and what makes cancer so bad? So cancer originates in healthy cells through the acquisition of genetic changes. This newly formed cancer cell is now able to divide and proliferate uncontrollably, giving rise to a malignant tumor. At later stages, cancer cells can even shed from the original tumor, travel through the body, and reach distant organs. And this is the real concern, because cancer spreading and the metastatic process is indeed what causes 90% of all cancer-related deaths. And that's why there's a huge worldwide need for the development of novel, more effective treatments that are able to control metastatic disease while reducing the severe side effects caused by available treatments that include nausea, diarrhea, skin rashes, fatigue, among others, in order to obtain long-term responses to treatment and thus improve patients' quality of life. Among the different genes that control cancer development and metastasis, there's a key tumor-promoting gene that is known for more than 30 years. When mutated, this gene stimulates cancer cell hyperproliferation, prevents cancer cell death, contributes to metastasis, and renders cancer cells less responsive to treatment. And these are the reasons why this key tumor-promoting gene has become one of the most attractive therapeutic targets. However, until now, there's no inhibitor available. And this is of paramount importance for both the scientific community and the pharmaceutical industry, because this key tumor-promoting gene is frequently mutated in several cancers with high prevalence and mortality, such as pancreatic cancer, lung cancer, and colorectal cancer, as well as in a rare form of cancer. And given the high frequency of mutation of this key gene and the large expertise of our team in colorectal cancer, we selected colorectal cancer as our initial target model. So colorectal cancer is a disease that affects 1.4 million people worldwide. Of these, 1/3 are non-metastatic and are successfully treated with existing therapies. The other 62% developed or will develop metastasis. And if we now focused on the gene that I've been telling you about, we can further divide this population into 2 different segments. So one that corresponds to metastatic patients that do not have the mutation, and because of that, they are responsive to existing treatments, living between 3 to 3.5 years. And a different segment, a second segment, that corresponds to metastatic patients that do carry the mutation. And because of that, they are no longer responsive to treatment, so they lack efficient therapeutics. And their overall survival is, at the very best, only 1 year after being diagnosed. But why are these patients not responsive to existing treatments? So to explain you this, I'll use the map of Metro do Porto as an example. So a cancer cell can use different pathways to divide and proliferate, just like the lines of Metro do Porto. And if you see here in this diagram, this network is composed of 6 different lines, but still there's only one central station where all of them converge and intercept. If we now make the analogy of this network to our patients with a mutated gene, this station corresponds to their mutated gene. So what happens with current drugs is that they block branch lines, and thus they allow cancer cells to still divide and proliferate via alternative routes. And thus, the tumor actually becomes resistant to the therapy or to the drugs. So what these patients really, really need is something that actually blocks all the circulation. So it blocks the central station in a way that it reduces tumor growth and prevents therapy resistance. And that's exactly what we have. So we have developed an innovative, patentable chemical compound that is able to inhibit this key target gene by more than 50%, as shown in their bar chart. If we now look at the viability of cells that are treated with our drug, you can see here that healthy cells remain unaffected, indicating the low toxicity and reduced side effects of our compound. But now, look what happens to cancer cells with a mutation. Their viability dramatically decreases by more than 50%, indicating that our drug acts in a highly specific and highly efficient way. And thus, it will be a unique solution for patients lacking efficient therapies. So this is our roadmap. We now finished the discovery phase and most of the in vitro studies, and we are ready to start the pre-clinical in vivo validation. We expect to reach clinical trials within the next 2 years, and at that point, either license the technology or be acquired by a big pharma, as happens with most drug development startups. Just this year, Impact Biomedicines, an anti-cancer drug, the startup, has been acquired by Celgene by the impressive amount of $7 billion US. And who is going to perform this? This great and amazing team of researchers, because we have multidisciplinary and complementary expertise, and we have people working for more than 15 years in chemical synthesis, cell biology, and oncobiology. And during this program, we have had the precious help of our MBA students and of our mentors. And we are here because we want to change the life of these patients. We want to give them the best therapeutics to control disease and improve their quality of life. And last but not least, I would just like to finish by saying that because we want to give this treatment to all patients in need, we are now starting or testing the efficacy of our drug into additional models, lung and pancreas. So thank you very much.